Hi guys, it's Miss Arneson, and I'm here to prep you for your final exam for Designer Babies Science 122. This is term 3 of biology, and there's two parts of your test question. But before I go into that, I want to tell you something really important that I did not emphasize yet, but which is going to be on your test. That's how you copy DNA and RNA. So normally, like if you put something in the copy machine and then you press the copy button, then you get basically the exact thing out, right? Unless it's a color copy and you're trying to print in black and white, and then it'll be like the translation of that picture into something else. So it'll be black and white instead of color. But when it comes to DNA and RNA, we're actually fitting puzzles of like pieces of a puzzle together, kind of. So you can't make an exact copy. This A shape right here represents adenine, one of the nucleotides that's found in DNA. And this one represents thymine. Adenine and thymine are going to fit together in DNA, and guanine and cytosine are going to fit together. I can always remember this because it spells at, and it also, the G and the C are basically the same shape. So that's how I remember that G and C go together and how A and T go together. So they don't fit together with another A. You can't copy this A into another A. What you have to do is make what's called a complementary copy instead. That means that instead of copying an A with another A, we copy it with a T. And so this still is going to carry the same genetic information because you're going to have two copies. This side will get copied, and then this side will get copied, and then those copies are going to go into the cells of the offspring. So either that'll be the offspring like of an amoeba, or it would be your somatic cells, the cells that make up the body of your organism. Your face cells get replaced with more face cells that also have your genetic information. So it's basically getting passed on. This is called DNA replication. And remember, we're making complementary copies. There's a question on your test that will give you a series of letters. So it might be A, G, T, and you will have to know that TCA is the copy that would be made of that for DNA replication. You also need to know the word DNA replication because it's going to ask you what copy would be replicated. If you had CAT, that's one of the few words that you can spell with DNA, proving that humans are really great because you can't spell dog with this. Cats are better than dogs, so humans are great because we can spell cat with our DNA, right? It makes total sense to me. So anyways, CAT would be copied with GTA. So maybe if you like Grand Theft Auto, it's in our DNA too, I guess. But anyways, you just need to know that that's called replication, and it's going to have A's, T's, G's, and C's. But there's also RNA, and RNA is a little bit different than DNA because it has one strand instead of two. So here's DNA, right? It has two sides. RNA is only going to have one side. Here's making the copy. And it's just like DNA where you can't make a copy of a T with another T, so you match it with an A because it's the right puzzle piece shape. But we're not going to use thymine at all in RNA. So we're going to use instead something called uracil, and we use a U to represent it. So here, if we were copying TAC, we would actually copy that with AUG. If we were copying TAG, we would have to have AUC. So basically, if you're making an RNA copy, you know that AT and GAC go together, right? It's guanine and cytosine, GAC. So you know those. If you want to put a T, but you're making RNA, you need to stop and put a U instead. Okay, so that's one thing you need to remember. This is called transcription. So I don't know if you guys know what transcription means, but if you had like, this is like you could be, there's commercials that say you could be a medical transcriptionist. And if you did that, basically you would listen to case notes from a doctor and you would type them out. So you're changing it from the audio version to a visual version or a written version. That is what you're basically doing here. We're transcribing this DNA version into an mRNA version so that it can go out and be turned into proteins later on. So let's zoom in at this process a little bit. Okay, the first question on your test is going to, the first short answer part of the question, you're going to need to write 2 to 3 paragraphs. I know if I was taking the test, I would need to write 3, but some people are a lot more brief than I am and concise. So we're going to talk about the 3 steps of gene expression. That's what it's called. Gene expression is how we get from DNA in your cells, which is this little molecule, to blue eyes or to black skin. Any of those traits that you have can be connected back to your DNA. But how exactly do we do that in our cells? I mean, we don't do it willingly, right? It just happens all the time, whether or not we like it. Well, that DNA can get copied or replicated in the nucleus. You don't have to copy your DNA all the time. You really only have to copy it before you're going to divide. So if one of your skin cells is dead and another skin cell is going to replace it, then it would need to copy its DNA. So the copies of the DNA really don't happen like all the time, like the copies of the mRNA do. But that's basically step 1. It goes DNA, RNA, protein, or DNA replication, RNA transcription, protein synthesis. That's the process of gene expression. And this part, the second set of pictures, is representing the RNA transcription part of that process. So what's happening is these little nucleotides are going and they're filling in. Remember, we have A's, T's, G's, and C's when we're talking about DNA, but we're introducing a new color over here, which is the orange, is going to be matching up now instead of the color that was representing thymine. So this RNA, after it gets copied, it's going to, and it's a specific type of RNA called mRNA, which stands for messenger, because it's going to take the message of the DNA out to the cytoplasm to get with the ribosome so they can actually build the proteins. So this part is called RNA transcription. Leaves the nucleus, goes and meets up with a ribosome. Now, ribosomes are these little proteins that float around in your cytoplasm. If you were making a cell model and you were like building the mitochondria and the nucleus, you'd basically have to just pour like those micro glitters onto your cell model to represent the actual scale or size of ribosomes. They're very, very small. And these ribosomes, even though they're small, they're really important because without them, we would have a good message, but we wouldn't have any way to have traits. So you'd have the genotype, but you wouldn't have any phenotypes at all. So we really need these ribosomes to build proteins. Proteins are built out of these little pieces, just like DNA is built out of nucleotides. Proteins are built out of amino acids. And there's about 22 amino acids, I think. And they're going to come and they're going to start getting put together, and they need to get put together in the right order. So of course, like Legos, you can put Legos together, and if you don't have a good set of directions or you don't have a pattern in mind, then you're just going to have a glob of Legos, right? So what's happening is this mRNA has a message of how to actually build like the spacecraft out of the Legos, so then you can have a cool Millennium Falcon instead of just a mess. And so these are going to be red. So you can see it's AUG. AUG matches with UAC. And so this tRNA is leaving, and it already left the amino acid right here. This tRNA is matching up with CUC, but it's GAG, and it's going to leave this one called leucine. The next one is going to match up with GGC because it is CCG, and it's going to leave glycine. And basically this process will go on until you get a whole entire protein. And then those proteins get put together into bigger things. And depending on, like, for example, if you have curly hair or straight hair, the curly hair has a different shape of like the, how do we call this? A hair follicle. You have like a different shape, and it makes your hair curl as it grows out. I have straight hair, so I have the one that doesn't do that. And so those are literally proteins that are in my DNA makes those proteins, and that's why I have straight hair. So this is the process. The second part of your question for your short answer is going to ask you about which part of the DNA, RNA, protein process CRISPR-Cas9 is going to modify. So when we change a gene or when we change an organism with genetic engineering, what part of this process are we changing? Are we changing the way that these amino acids are going to be put together? We are, but the way we do that is by snipping out a part of DNA and putting something else in there. So Cas9 can either just like delete one of these, or it can cut out a whole entire piece and put in a new piece even. So we can really change basically anything we want about the genetic information. The reason that we're not making designer babies right now is just because we don't have enough practice to do it. But in your lifetime, I'm pretty sure we're going to have designer babies. So you're going to be voting based on what you believe is right or wrong for genetic engineering. And if you go back to one of your assignments, hold on, let me see here. One of our assignments was about all the things that could happen with genetic engineering. And we talked about how like humans could basically make slaves genetically. So if they wanted to, I mean, humans have enslaved people for most of history. So they could basically make slaves that do what they want. They could have like an army of people that fights for them, but they could genetically engineer those people to be aggressive and to take orders and not be questioning and to have bigger muscles or better eyesight so that they could fight more effectively. There's also the issue of cost, right? So right now, I know in the United States you get a free education no matter who you are, but if you go to other parts of the world, they really don't get a free education. I had a student who was one of my best students I ever had in class, and I asked her why did she love school so much, and she told me, well, because I used to live in Mexico and we had to pay for school, so I wasn't allowed to go. Like I didn't go to elementary school because my parents couldn't pay for it. So those are things that might happen with genetic engineering too. So if your parents can pay for you to be better looking, maybe you're going to get a better job than someone who has just as much smarts as you but isn't as good looking. Or we could pay for people to be able to manage their weight easier, or you could pay for people not to have a risk of diabetes. And so all of these things would be in demand, but they would end up costing money, and who should pay for it? Who should get these things? So here's a list of things that, remember, I asked you to look at and decide if you thought they were right or wrong. You can use these ideas here to decide what you think should be done with genetic engineering. So the last part of that question, that short answer question, first you need to describe how does DNA get made into traits in the first place? How do we get these genotypes into phenotypes? Then you need to describe how CRISPR-Cas9 is a genetic engineering technology that basically cuts pieces of DNA out and can insert new ones so that we could change traits in organisms. But these genes could also be passed down. So if you modify a baby before it's even born, all of its cells in its whole entire body are going to have the new DNA. Is that right? Should we be changing something that's going to get passed down forever if they have offspring? I can't tell you if it's right or wrong. You're going to have to decide. And that's what you're going to write in your last part of this short answer question. What is right or wrong? So you're basically going to say, how does it happen naturally? How have humans figured out that we can change it, we can engineer it, we can make it however we want? And then the ethical decision of what we should or should not do with this new technology. So that last part, and in science a lot of times I don't get to ask you for your opinion. In history, like all the time, Mr. Arneson gets to ask you for your opinion, and he lets me read the answers, so then I see what you guys are thinking anyway. But I don't get to ask very many cool questions like this that are not just about like, well, what happens, but what should happen, what should we do? And so I'm really looking forward to reading your answers on this because I love them. You guys think of things I never would have thought of, and I've thought a lot about this, and I've watched a lot of sci-fi, so I have a lot of ideas about what could happen with genetic engineering, but you guys always come up with new ones. It's one of the beauties of being a teacher, is I get to learn from my students as well. So if you have any more questions about this short answer section, you can ask me, but my best advice would be, while I was just talking about all the things that need to go in there, you can write some of those things down, make those into your short answer, and then all you have to do is figure out which your opinion is, which I can't tell you, of course, right? I can't tell you what your opinion is on what we should do with genetic engineering and why we should do that. Okay. Dang. I'll see you guys later.